So this is all review session, and let me say a few things up top here. So I have a pretty good solid draft of the exam 4, and I'd like to give you feedback on how to rank out like which topics have the most points, working forward. So number 1, the number 1 point value, most points are coming from this slide set on the C4 cycle and the CAM cycle. So far, that's what we have ranking out as number 1, most points. And then the next highest point value coming from the slide set on mechanisms of plant defense. So when we were looking at how does a plant figure out how, who's attacking it and then what does it do? So we'll get into that slide set as well today, of course. So that's number 1 and number 2. And then thinking about the slide set on signal transduction and an intro to hormones, looking at how hormones are used as signals. So that slide set, and then I kind of, if you combine that with the applied uses of hormones, that slide set, those two combined would rank out to be the 3rd most point value. So the signal transduction, intro of hormones, and then the applied uses of hormones, those two slide sets, when you add up the points, it's 3rd. Then the next ranking would be secondary metabolites, followed closely by signals from sunlight. Those are about equal, actually. Signals from sunlight. And then the different abiotic stresses. Drought has the most out of all the stresses. There's more points coming from drought and heat. And then chilling and freezing and the flooding are about the same point value. They're ranked the least amount of points. Got that? Okay. So that should help you strategize your focus for Monday. Yes. Thought I had that. I think it's around 45. Might be closer to 40. And so right now I'm at the stage where I tend to do this. I'll make the test and then I cut out stuff before I say I'm good. So I'm guessing it's going to come down a little bit. It's a little bit over 100 points right now. I'm guessing it'll get closer to 100 or maybe even below, a little bit below 100. It'll hover around 100, I'm guessing, by the time I'm done. So I'll be doing that over the weekend. Takes several days for at least the way I do it. It takes me several days to get the final. Other questions on just the general ideas here? So remember, it's no essay, it's multiple choice, true, false, drag and drop. There's no fill in the blank or any other thing that I can think of. So, and please remember, just like with all the other exams, even though we don't have an essay portion, I still, any question that's confusing to you, make sure to ask for clarification. All right, so don't be afraid to ask questions during the exam. I'll come around and help as much as I can. So with this slide set, remember the first half of it is gone already. We already did that. So it's essentially slide 40 onward that we're thinking about for this material. So we went through this a little bit on Wednesday already, but are there any questions lingering on C4 or CAM? Because that's the top point value. C4. Any questions on CAM? Yeah. With respect to C4 and CAM, like why the heck did plants even bother evolving this strategy? What's the point? So that's how I would get you to think about the PCO and what, so generally think about the big picture idea of the PCO and why we generally think that's not such a good thing to do on a regular basis. So just the basics of that, and then put it into context of, well, why would these plants bother to evolve C4 and CAM? Yeah, so like why, if you look at the C4 or the CAM, both of these images kind of show the idea of the benefit to running the C4 or CAM. Even though there's an extra energy cost, they still keep doing it. They don't get rid of this strategy through evolutionary time. It's persisting. But why? What's the advantage? Thinking about the PCO, specifically, what's happening in this cycle? Correct. So so with the PCO cycle, why does the PCO cycle even start running? There's something going on that triggers the PCO cycle. Rubisco can't distinguish between CO2 and oxygen. That's the crux of the matter right there. So what does the C4 and the CAM do? Removes the issue.
C4 like separates the different cells.
Yes. Yes, you have this strategy of, in this case, it's a physical separation, so that when CO2 first comes in, it doesn't even react with Rubisco. It reacts with peptase, which is super strict. It doesn't care about oxygen. And then as this percolates through, keeps going, see that CO2 is kind of swamping or bathing Rubisco. And so if you keep this going, so it's kind of a multi-benefit here, because you have C4 plants, they use peptase in the mesophyll cell, and there's plenty of oxygen here. The mesophyll cell, plenty of oxygen. And yet peptase is like, yeah, I don't care. I'm just going to take CO2 in. So that's great, because then the plant gets the CO2 attached and then malate, and use malate to deliver it to Rubisco. And in the bundle sheet, Rubisco, you don't have a light reaction going on. So there's not a buildup of oxygen. And so now Rubisco is just getting lots of CO2. So that strategy is really good. The peptase enzyme, and then just bathing CO2 or Rubisco with CO2. So peptase and malate. Pardon? Peptase and malate. Yes, peptase can as well. Yes, and malate. Yes. Malate, malate, malic acid, all kinds of names for that one. Other questions on C4, CAM? Okay, well, if you think of something, we can come back, but let's go into the next slide set on mechanisms of defense. That's the next top thing. Is it this one? Yeah. Okay. So this slide set is getting at some of the big picture ideas of how do they defend themselves toward the start here. And then we start looking at some terminology. So you need to know the terminology, like, when we talk about elicitors, for example, that idea, I'll expect you to know what that means. So unfortunately, there is a fair bit of terminology and acronyms. So, like HAMPS and MAMPS, we talked about those. So we kind of looked at overall general ideas, and then we started giving some examples, like what happens when you have, you know, the caterpillar, in this case, chewing, you know, what's going to happen. And so again, it's a process where, you know, first the plant has to figure out, oh, I'm being attacked. And then it has to go, okay, based on the signals it's getting, and it has to discern what am I being attacked by. And then it has to kick into gear the right response that will hopefully allow it to survive. Right. And so we looked at like this idea where we have a caterpillar here on the tomato leaf. And then they're trying to show this kind of step-by-step process of, you know, the systemin protein, which is actually a hormone, is it kicks off, and then that kicks in the jasmonic acid. Jasmonic acid can course through the plant to all the other leaves. You know, so it's kind of the step-by-step process that I'd like you to think about. So we have kind of what happens when there's insects chewing. We looked at those ideas, and then that it's not just chewing. There might be vibrational issues that they can pick up on. So we talked a little bit about that. And then we started getting into the idea of the plant can warn nearby plants. And then some of the other subsequent slides further down, we get into more of, okay, if a plant's getting attacked by a fungus or a bacteria, what does it do? So it's kind of like insects and chewing organisms versus, excuse me, then we got into the pathogen ideas, looking more at fungi and bacteria, the MAMPs. This slide, we looked at how it's comparing two different versions of immunity. So I will be talking about, you know, MAMP triggered versus effector triggered. So knowing the difference, what's going on with those. Questions on anything in this slide set? We went into a lot of what's happening here. We talked about all of that in the subsequent slides. So we looked at the hypersensitive response, what are phytoalexins, the lignin response, hydrolytic enzymes. Questions in this slide set? Yes. Good question, because I think I said this when we were covering it initially. Please get those two straight in your head. Please, please, because I will be asking you what's happening during systemic, what's happening during induced. So please get those straight. So systemic acquired, it is the plant initially figuring out it's getting attacked. And then it's the plant's ability to respond appropriately to that attack, plus warn the rest of its body that it might be, you know, the other leaves, for example. If a caterpillar is chewing on a leaf, it's got to warn the entire leaves of its body, hey, you might be next, ramp up. And so that's the systemic idea. Even though it's, you know, maybe just one caterpillar on one leaf, the plant can figure that out and send this whole signal systemically so that resistance builds up throughout the plant body. The other payoff is that it kind of creates this memory where if you challenge that same plant with the same exact caterpillar, maybe a month later, it will have the ability to respond much more quickly. So it has that elevated defensiveness, much like how our memory T cells allow our body to have that elevated defensiveness. So those are two key points of systemic, but it all is kicking off with the plant, you know, the plant is the one saying, oh, I'm being attacked. Induced, oh, let me finish the thought. And then systemic, I will be asking, like, okay, so during the systemic process, what hormones is the plant using? Because the plant will be using separate hormones depending on if it's a systemic acquired or induced, different pathways. And so systemic, salicylic, methylsalicylate, the methylated version is volatile, so that can help with distant leaves, perhaps, or neighboring. So I like this one. I think that's a nice visual that really kind of encompasses everything with that idea. So that's systemic acquired. So induced, the way I guess I think about this is to induce something, it's like something has to trigger it, something else besides the plant. And so you're inducing the plant. And so it's an outside entity that's inducing the plant. And so in this case, we're talking about beneficial microbes hanging out, coexisting with the plant. And they're actually capable of assessing that there's something going on with regard to a pathogen, and they can actually trigger the plant to start defending itself. So it's the bacteria inducing the plant's defense system. And it'll still go systemic. The whole plant can get ramped up. But it's because of those nearby bacteria, or maybe they're endophytes, maybe they're inside the plant, but it's some microbe, bacteria or fungus. So, but they do it through a different route so that the plant can tell the difference. All right, so it's not salicylic acid, and it's not the pathogenesis related proteins that or genes that are going to get ramped up. Instead, they use a different pathway so that the plant can figure it out and tell the difference. So jasmonic acid and ethylene are the difference here. So different steps. And then this is showing that idea here, where they're trying to show the beneficial bacteria, telling the plant, hey, you better gear up. Something's about to attack you. Potentially, but usually most plants will defend themselves by ramping up secondary metabolites. However, I mean, sometimes you'll get so much lignin, extra lignin laid down that I've seen some reports where they'll like then upsize off the leaf. Like once it's like, okay, it's just, we got to get rid of this leaf. It's just chock full of pathogens. And so sometimes they will go through these decisions. But for this purposes, we focus in more on the secondary metabolite idea as. Oh yeah, ethylene could cause that. Yes, for sure. Yeah. So ethylene does have a role in leaf abscission. Yes. And that can happen if it gets bad enough, where leaves will get sacrificed if they're too infected, for example. But I didn't really get into that for this. So. What other questions on this slide set? Whoops. Well, okay, let's shift into the next one. So that would be signal transduction, intro to hormones. So signals, well, not signal transduction. Okay. So when we were looking at this originally, I said, you do need to know like the basic flow of the process. So that's a good one to look at for that idea. So this is kind of a general overview of the process here, and then examples, like, okay, what's happening in each step of the way. So this is a good slide to kind of think about it in broad terms, like what's the big picture idea here. And then we started looking into some of the, examples of each step of the process, what's going on. So of course, when you have some kind of signal in the environment, the typical strategy for the plant is to have some kind of protein receptor somewhere that it can interact with that signal. Many of them are on the cell membrane, but not all. You have several receptors that are definitely internal, and it just depends. You see a variety of options here, the endoplasmic reticulum, or even inside the nucleus, chloroplast. So there's options. And so the plant has to have receptors for the signal. And then we started saying, okay, but that's not the only thing they do. They have to often like amplify or ramp up the signal. And so those second messengers are playing a role in that, where that is helping to kind of amplify and transmit the signal in the cell. And then, this slide, we didn't get into details. I was just having you kind of zoom in on the idea of the yellow balls, acting as calcium. I'm just trying to get you to think about how they're, being sent into the cell and shifting. We also have pH gradient shifting, so those are protons that are moving around. That also can act as a second messenger. So the plants are paying attention to all of that and using that as a way to further kind of refine the signal and amplify it. Even cell membrane lipids that break off from the membrane. So like if a caterpillar is chewing and some of the membranes get destroyed and the membrane lipids are floating around, that's a signal, a second messenger. And that the plant can go, oh, okay, I have just 3 bits and pieces of membrane, and that can help them ramp up appropriately. Now, I just mentioned calmodulin as a protein that interacts with calcium, but I'm not going to ask specifically. And then, of course, hormones. We got into the idea that you have hormones as message, helping to relay the message. And so then we just talked in general terms about, here you see the signal, the environmental input, that's just kind of signal from the environment, often triggers different hormones to get made. And then they have to get to the right part of the plant body and dock into a receptor so that they can relay the message to the cell. And eventually there has to be a way to turn all that off. So eventually that signal has to get downplayed. So that's what they're showing on the right side here. Like, okay, once the plant has hopefully responded appropriately, eventually it can't always be doing that. It needs to kind of shut that down. And so there will be different ways. So there is this dynamic of feeding back and saying, okay, we're good. You can, quiet down all of that. So we looked at the different hormones in general and said a few things about them in this slide set. Auxins, of course, very important for the plant. So some of this material, we looked at some of these hormones in lab as well. So some of that material might sound familiar. So knowing the basics of what does auxin do for the plant? What does cytokinin do? So coming off the slide set here, gibberellins, we looked at them as well. So I focus mostly on the classic five. So that's the auxins, cytokinins, gibberellin, ethylene, and abscisic acid. Those are the ones that I'll be focusing on when I ask you specific questions about hormones. Questions on anything in this slide set? Yes. So yeah. So one of the main points I was trying to get at with this, with talking about reactive oxygen species, is that in like general, like non-science resources, they tend to treat reactive oxygen species as bad, bad, bad. And I wanted to kind of correct that. In the plant world, reactive oxygen species, yeah, it can be bad for the plant, but they use it as a signal too. It's a way to figure out they're getting attacked. And so reactive oxygen species that the plant makes is actually, yes, it can be good, and yes, it can be bad. And so if it's good, it's actually allowing the plant to respond to a stress appropriately. On the other hand, if they get too much reactive oxygen species just by like, free radical formation through too much light energy, and oh my gosh, now we have a bunch of reactive oxygen species, then it can cause damage. So I guess that's one of the big things I wanted you to grasp with that the plant is using reactive oxygen species as part of their stress response or their defense response. No, I won't ask specific. I'll just say reactive oxygen species. So I'm not going to ask. I just put this in here to give you some examples. Those of you who are good at chemistry were, oh yeah, those are free radicals. Not so good. And then I just think it's interesting that that's clicking along in our bodies right now. In our bodies, we do have frequent production of free radicals, but hopefully our bodies are handling it well enough. Whoops, what just happened there? Okay, accidentally shut it all the way out. Whoops. Other questions? Okay, so then if you combine this slide set with the applied uses, so these two I kind of lump together because they cover some of the same stuff. So this one is looking at like how our knowledge of hormones now being used in commercial purposes mainly. So what can it do in that regard? So again, just some characteristics of the different hormones and how they've been applying the hormones to get certain qualities that they want in the plant or allow for certain things to happen that makes it efficient. And so again, going by what does auxin do? How are they using auxin in the real world? And so looking at those types of ideas. So if, for these types of questions, my question will have multiple cues, hopefully. So I usually say, which hormone does this, 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 and this? So I'm hoping at least one of those will go, oh yeah, I remember. All right, so I don't just say one thing. I'll phrase it so that there's at least two, if not three, if not four things that I'm telling you that this hormone does. So it's a little bit easier to trigger your brain cells. Any questions on anything in here? Well, what about secondary metabolites? So we just looked, there was a fairly succinct slide set. Just so, you know, being able to think about the three classes, of course. Which one has the most, so far, most compounds of the three different classes? Which is the biggest class? Do you remember? I'm sure you can learn this in your chemistry classes too. Which one? Terpenes, phenolics, or nitrogen? It's actually terpenes, so far anyways. More terpenes out there. Although I'm seeing a lot of interest in, well, at least in human health, there's a lot of interest in phenolic compounds. Polyphenols, they're really zooming in on those for human health. But anyways, what kinds of questions do you have on this slide set? You want to see the let's see. I don't think I mentioned coumarin. I don't think that one's in there. So, let's see. Like flavonoids, think about the flavonoids. They're in there. I guess, yeah, tannins are on slide 13, I think. Essential oils are fun to think about. I'll have that in there. And then a fair bit on the nitrogen containing compounds. Oh, and yeah, and I do ask a little bit about endophytes. I think there's at least one question in there, maybe two, on endophytes in general. So I put those few slides in there because I want you to realize that plants are not the only ones making these secondary metabolites, that there are the associated endophytes inside that can do the same thing, or maybe different. And unfortunately, plant physiologists didn't discover them until pretty late in the game. But at least now they know they're there, and they're finding that they can be incredibly helpful for the plant. And lots of commercial application for that as well. So good to be aware of. Any other questions on this slide set? Yeah, so let's see. No, so signals from sunlight is like close to how many questions are on, or points are for secondary metabolites. So I'm going to do that one next. So secondary metabolites and signals from sunlight are kind of running neck and neck right now for point value. So here, this is signals from sunlight. So there's not a whole lot here, but you can bet I'm going to ask about phytochrome and just in general, like what is a photoreceptor? And then phytochrome. So are there questions on phytochrome? Historically, I see, students get confused on this, so please ask. Well, cryptochromes are a totally different beast. Phytochrome, one of the claims to fame of phytochrome is that it can toggle back and forth. It's reversible. So in here they're showing here where it says red light goes off to the right, far red light goes off to the left. And so it can toggle back and forth to these two different physical forms. So it's kind of, at least so far, that's unique to phytochrome. And then when you get into cryptochromes, I mean, they're totally a different beast. It's very different chemical composition in that regard. And they deal with blue light, whereas the phytochromes are zooming in on red and far red. So questions on phytochromes. So these are pigments, but are proteins. But when you look at the trying to think of what some of the common misconceptions are. So the piece of art, the reason that they put the substance on is that what they mean is when it's in that conformation, it's ready to absorb red light. And so piece of R means that it's the version that responds to red light, absorbs red light. So then if piece of R is getting exposed to red light, what it does is it physically shifts its form into what they call a PFR form. So I know it's confusing. When I first had to learn this, it was confusing. So the piece of R form is ready. It's like designed to absorb red light. That's what it responds to. That's why they put the R there. And so it'll absorb that red light, physically shift into what they call the PFR form. And then that form is designed to respond to far red light. So if for some reason now this piece of FR form gets exposed to far red, then it's going to physically change its shape back. And it's the ratios of these. The plant's going, how much piece of R do I have? How much piece of FR do I have? And it keeps assessing that ratio to figure out what it should do, how it should respond. So if you're a plant that is living, if you're a giant oak tree and you're at the top of the canopy in an ecosystem and you're trying to crank out photosynthesis and make your sugars, what quality, what color of light will you preferentially grab? Red, right? So for photosynthesis, if you're in a forested ecosystem, the top plants got it made because they're like, I'm going to grab and absorb the red light. Well, if they're absorbing red light, that means red light's not really getting past their leaves. It's not getting down into the understory, at least not very much. And so, I mean, some gets down there, but in general, the lion's share of red light is taken by the top canopy. So that if the red light is being absorbed by the top, that means the far red light is coming down into the lower canopy. That's what the lower canopy gets most of. And so you can see how, the plants in the upper canopy, that giant oak tree, if it's absorbing most of the red light for photosynthesis, but it's also absorbing it and it's interacting with the phytochromes. And so that giant oak tree at the top canopy should have a high level piece of FR in its leaves because it took it hogged a lot of the red light in that ecosystem. Does that make sense? So then the lower plants, there's a fern on the forest floor below that, it is going to be getting hit with far red. So that fern on the forest floor, it's going to have a very different ratio of these phytochromes because of that compared to the oak. It's going to have way more piece of R. So, please spend some time with this because there's at least 2 questions, if not more, on phytochrome. So, and I remember first learning it, I had to slow my brain down, really think through this. So I would encourage you to like sketch this out, talk it out. And then, so phytochrome definitely is of the pigments. That one's the most important. And then we just knowing in general, like cryptochrome's a blue light one. That one handles blue light, and some of the things that it might be capable of doing, and you know, in terms of what does it do for the plant. And let's see, phototropins. We looked at those. And then toward the end, we looked at the ability of plants to even deal with ultraviolet. They'll use that and have the photoreceptors that can deal with that as well. So that was kind of a surprising find. They didn't really anticipate that. Any other questions in this slide set? Okay. And then the other topics on abiotic stress, drought. Of the 3, drought has more points coming. That. Oh, that's chilling. Where's drought? Here's the one showing how drought, sometimes they can amputate leaves or even more. What do they do in terms of often you'll see a differential response, like something will happen with the roots. That's the opposite of what would happen with the shoots, for example. So that's why when we do biomass measurements, we often, almost always, we separate out the shoots and the roots because very often the plant will respond differently with its roots versus its shoots to whatever's happening in its environment. Questions on any of the drought issues? So thinking about what does that do, of course, to the soil, but then getting into how does a plant deal with it? They actually have a fair number of strategies to a point. Unless they get to the permanent wilting point, then they're pretty much done for. Yes, so you know, morphological changes, the physiological issues that they'll try to employ, the hormones. We talked about abscisic acid is heavily involved in having a plant deal with conserving water. Questions on this? A couple of questions on chilling and flooding. Some of the terminology, acclimated versus adapted, know the difference, and like what can a plant do physiologically to try to deal with it? Abscisic acid again involved in this issue. Any questions on chilling, freezing?
We should probably.
Yes, the saturated versus unsaturated, important concept. And then flooding. Basically, we have two things going on here. We have, of course, flooding is messing with the ability of the plant to get oxygen, but we also have a lot of stuff going on in the soil in addition to that, that is causing issues for the plant. So know what, both what's happening in terms of the plant is really having issues with oxygen availability, but then we have all those issues in the soil, mainly due to microbes causing problems. You get the wrong types of microbes starting to overpopulate, and all their activities cause even more problems for the plant under flooding. So the plants do have some strategies, some pretty neat strategies actually. It's amazing what they can do to deal with flooding. There's only so much they can do. Questions on any of the slide sets? Anything right now? Okay, I'll be working on the weekend again, so if you do have like some confusion and want a quick response, like, hey, I'm confused on this, send me an email. I'll be checking email all weekend, so just let me know. So the exam's Monday at noon in here. Today, yeah, the plant sale I think is still going on today, all the way till I think 4.